Hello guys, this is complementary strand. First, we hit the strand. This is the template strand. This is primer combined with template strand. Template strand is added to the test tube. After that, DNA polymerase is added into the test tube, followed by all other 4 bases of DNTP. And last one, the bases of DNTP is also added. DNA polymerase is now attached to primer and consequently seal all the bases together. Now, the mixture of first test tube is added to the first column, second test tube added to the second column, and so on in the gel electrophoresis. After inserted into the electrophoresis machine, it will produce a different length of strand. This strand will arrange in sequences which produce complementary DNA. And finally, a new complementary DNA strand will be formed. So let's dance!
DNA sequencing is a method used to determine the nucleotide bases A, T, C, and G of DNA. Every organism on the planet Earth possesses a complete genetic material. The genetic material contains the full genetic information about the organism. DNA sequencing allows the scientists to use the genetic information in different purposes such as medical research and forensic. Certain biological processes also take place because of the genetic information obtained from the DNA sequencing. DNA sequencing techniques have made possible to do the sequencing of human genome.
Here are the few examples of the application. First, in the forensics field, it can be used to identify particular individual as every individual has unique sequence of DNA. It can also be used to identify criminals by finding proof from crime scene. Next is the medical field. It can be used to detect genes which are associated with some heredity or acquired disease. And the third, agriculture field. Mapping and sequencing the whole genome of microorganisms allow agriculturists to make them useful for crops and plants.
The benefit of DNA sequencing. DNA sequencing used to determine the genome sequence. The biggest example is the Human Genome Project. After the project, we can identify the genes which are responsible for causing genetic disease. For example, Alzheimer's disease, cystic fibrosis, and myotonic dystrophy. Besides that, we can also identify the acquired disease like cancer.
The common challenges of DNA sequencing with the Sanger method include poor quality in the first 15 to 40 bases of the sequence due to primer binding. The length of DNA being sequenced cannot be longer than 1000 base pairs long, or it will be completely inaccurate. In addition, this technique of sequencing has another major flaw as it requires that the sequences be read by a person. Lastly, the most significant burden of the Sanger methodology is the cost.